The document that I'm going to be scribbling in the video would be provided through a link in the description, and also the link would be there in the first comment. Please do check it out, and let's get on with the video. Okay, so there is another level of regulation that goes on in the lac operon. In switching on and off the lac operon. Now, like I said, lactose needs to be utilized when there is no other, when there is no glucose in the medium. So the lactose which is present, it will be used as a carbon source by the bacteria. Now, holding on to that thought, when glucose is low and lactose is high, when this condition appears, glucose is low and lactose is high in the medium, there is a molecule inside the cell, inside the bacterial cell, which is called CAMP, cyclic AMP, which is the cyclic form of adenosine monophosphate. Now, this cyclic AMP concentration, it sort of increases inside the cell when glucose is low. So, you can relate these two. Glucose is low and cyclic AMP is high. So when cyclic AMP is high, there is a protein called CRP, which is called cyclic AMP response or cyclic AMP receptor protein. Cyclic AMP receptor protein. So this CRP, it basically binds cyclic AMP. So cyclic AMP comes and binds to the CRP. So if I draw it over here, if this is CRP, then cyclic AMP would come and bind to CRP. And together, this cyclic AMP and CRP, it causes positive regulation of lac operon. That is, cyclic AMP CRP, when they're together, they will go and they will bind over here. They will go and they will bind near the promoter. They will bind near promoter of the lac operon. Once they're binding near the promoter of the lac operon, binding of cyclic AMP CRP, it will facilitate more strong binding of RNA polymerase. So binding near the promoter, the cyclic AMP CRP, it causes strong binding of RNA polymerase. And once the RNA polymerase binds, it is going to, once RNA polymerase binds with greater affinity, it is going to transcribe all the structural genes, and eventually they are going to be encoded into the proteins that they should be. Now, when glucose is high, then the cyclic AMP becomes low. Now, when this happens, this whole process of binding onto near the promoter and with greater affinity, the RNA polymerase is binding. All of these is not being held. So when glucose is high and cyclic AMP is low, we do not need to synthesize, the bacteria do not need to synthesize the structural genes that are responsible for the lactose metabolism. And hence, when glucose is high, it will utilize the glucose. Hence, it will reduce the concentration of cyclic AMP, and therefore, it will negatively regulate the lack operon. So over here, it is going to negatively regulate lack operon. Right, so these are another two levels of how the lack operon is being regulated by cyclic AMP and another molecule called CRP. So CRP, sometimes it is also called CAP, that is catabolite activating protein. So catabolite activating protein. So this is another term that is used for CRP, which is called CAP. So the CAP binding portion is somewhere near the promoter, so that it can bind the RNA polymerase with greater affinity onto the promoter, and that is going to cause the expression of the structural genes more rapidly. So that was all about lack operon, the overview and the basics and how it is regulated. This note that I just made while explaining, this is going to be available in the description. There would be a link in the description where the note is going to be available. You can just click on the link and you can download this document and you can refer to it while you're studying. And I'll see you in the next video.